We've already mentioned this in another video, but scientists have much evidence for evolution. Evidence that goes beyond the fossil record, which we already talked about, radiometric dating, stratification and erosion rates, climatological evidence such as tree rings, sediments, and ice cores, fossil evidence as well, and it's things like index fossils. In addition to that, you also have molecular biology, which can compare analysis of DNA sequences and actually figure out how old the splits of the tree of life actually are, with a process that we call molecular clocking. But in addition to all of that, evidence for evolution comes for analysis of similar structures in different species. And remember, looking at morphological and physiological similarities, we can find out things like analogous, homologous, vestigial, and mosaic structures, which will tell us about the history of the evolutionary process. Remember, analogous structures are evidence of convergent evolution, such as what you see in the wings of birds, insects, bats, and reptiles, such as pterodactyls or ancient dinosaurs. All of these organisms, sometimes some of which have lived in different histories of the Earth, all have the same anatomical feature because they are under common pressure, showing again that evolution is a thing because when the pressure is similar, similar structures evolve to deal with that pressure. Likewise, homologous structures, or similarities that exist because animals share common ancestors, tell us about the evolutionary history because it tells us that we are alike other organisms because we share common ancestors with them. For example, the bone structures of different mammals are so similar because we all come from the same ancestral mammal. So even though those bone structures have been modified as we diverge from each other in what we would call divergent evolution, we still have much of things in common because we share these common ancestors. A special type of homologous structures called vestigial structure is a structure that we have left over from our ancestors. For example, the whales have a vestigial pelvic bone that they no longer need, and we have things like appendixes and muscles behind our ears and larger brows because of the things that our ancestors had. So this is evidence of how our ancestors used to be like because it is not a feature that's useful for us. And why would that be on us if we were designed? On the contrary, they're evidence of previous steps of the evolutionary process which are left over because they're not yet devolved or not yet disappearing from our structures since they do not present necessarily a disadvantage. So they're left behind. And we also have mosaic structures, which are structures which evolved from one purpose but end up being used for another, like human hands, which evolved from limbs which were used for ambulation. And that's why human hands are not perfect for grasping. They're great tools for grasping. They have opposable thumbs, and they have, other features which allow it to become good tools to grab things, but they're not perfect tools to grab things because they came from tools which were used to walk. And mosaic structures are, again, evidence for evolution because it tells us that the features that we have are not perfect for the text that we have because they carry the legacy of what this used to be. Remember, descent with modification. That means no structure in no organism is perfect for a task because the structures are coming from a previous existing structure. It's modified structure, and that means that it's going to have limitations because of that. And remember, all of these structures that occur at the anatomical level can also be observed at the molecular level. There are similarities in the genetic code which indicate common ancestry, homologous DNA pieces. There's also similarities indicating common pressure, which are analogous DNA pieces. Pieces of DNA which are left over from before, which are no longer useful, called pseudogenes, which are an example of vestigial DNA structures. And you also have DNA sequences which evolved for one purpose but are used for a different one, which we call mosaic DNA structures. So this is also true at the molecular level. Other evidence for evolution comes from biogeography, which we see repeatedly in microbiology, virology, mycology, botany, and zoology. Microbiology, again, is the study of microorganisms. Virology is the study of viruses. Mycology is the study of fungus. Botany, the study of plants. And zoology, the study of animals. But what we see across all of these studies is that there are patterns of diversity in the world across space and time, and that organisms seem to be matched to their environments and to the pressures of their environments. And that's because the organisms which do not have the set of adaptations that allow them to survive will die off, leaving behind only the organisms which can survive better in that environment. And that means that over long periods of time, the species will actually adapt to the environmental constraints that they have. Remember, this happens over generations. It's not the same thing as you adapting during your lifetime. We're talking about evolution here that occurs through successive mutations which are selected for or selected against in addition to random processes like genetic drift and things like that we talked about in other lecture series. So studies from biogeography tell us about these patterns of diversity and provide even more evidence for the theory of evolution. These are all examples that we've already talked about, like antibiotic resistance, like viral resistance to medication, pesticide resistance, and examples like the cichlids in the lakes or the guppy fishes evolving in different pools where the pressure was different. We also talk about macroevolution evidence where we see actual species becoming other species. And there's also evidence from embryology or similarities in the development of embryos and how the embryos share common ancestry. We can see that in the embryos because we are all pretty much very similar until different genes which are only present in certain animals activate. For example, in the beginning we're all the same, but as the genes which are only reptile genes activate, then that differentiates from the fish embryos, and then the genes which are mammal-like genes activate, and that differentiates from the reptile embryos. But in the beginning, fish, reptiles, birds, and mammals, the embryos look very similar to each other because we have common ancestry, and until those extra human genes activate, we will look much like the previous existing versions of life. And it's a great way to actually see the history of the development of life, because we have leftover features from our ancestors in the embryological stages of development prior to a deactivation of the genes which are only in our kinds. You also have evidence from parsimony, because evolution is the simplest explanation that can actually fit all the data that we have. So when we put all of this data together, we can understand that evolution is the best explanation for the nature history of life, and that's why we actually have to accept the theory of evolution as what explains the natural history, the diversity, and the forms and functions that we see in life today.